Welcome to our video lecture on transcription and translation. Our essential understanding for today is that genetic information in DNA can be translated to make the proteins needed by the cell. Our objectives are, we are going to define transcription, we're going to describe that process of transcription, and we're going to talk about some of the ways that we can control what genes are transcribed and which are not. We'll then define translation, talk about the structure of a ribosome because this is what catalyzes those chemical reactions in translation. We'll practice using a codon chart. We'll describe how translation even happens. And then very briefly, we'll comment on how the genetic code is universal. So transcription defined. Transcription is the synthesis of messenger RNA, mRNA from the DNA-based sequence. And we're going to make that happen with RNA polymerase. So this is the enzyme that we use to make messenger RNA from our DNA. Interestingly, only about 1% of our human DNA actually is transcribed into proteins. The rest of it, that other 99%, we originally called junk DNA because we didn't know what it did. But we've actually learned a lot in the last 30 years or so, and we know that a lot of that DNA does a lot, but we don't know yet everything that it does. So transcription, the first step, RNA polymerase, that enzyme that is going to carry out transcription, polymerase is going to bind to the DNA at what we call a promoter site. And the promoter site of a gene is just this starting space, a little bit of a DNA sequence that is going to attract that enzyme RNA polymerase to bind to the DNA so that it can turn a gene into messenger RNA. We can actually edit, control how that polymerase binds to promoter sites by adding different molecules to what we call nucleosomes. So nucleosomes are the histone proteins. They usually come in groups of 8, so an octamer of histones. And the DNA, this is the DNA that wraps around that histone octamer. The combination of that DNA and those histones is known as a nucleosome. This is another picture, same idea, but now we're just looking at it more from a molecular standpoint and looking down at the top of it. So like this view, you can see the protein, all those beautiful alpha helix secondary structures in there on those histone proteins. And then here's our double helix DNA wrapped around it. So we can take some other molecules and we can add them to our histones and DNA, add them to our nucleosomes, and we can use those extra molecules to either add more access to the promoter site or we can close it down a little bit and turn it off. So this heterochromatin forms when we have methyl groups that we add to the nucleosomes. So these methyl groups, just molecules that I have tacked on to our nucleosomes. So these methyl groups, they really like each other. And so they will pull, they will pull the molecules of nucleosomes closer to each other. And if I have a promoter site like here, it's going to be really hard for my enzyme. Here's my enzyme. It's really hard for him to get in there when things are all squished up. So methyl groups, methylation, will result in very little or even no transcription happening on that gene. That's different from acetylation. So if I add an acetyl group instead of a methyl group, these acetyl groups actually push each other away a little bit, and so we open up that space. If my promoter region is here, it's super easy for my enzyme to come and attach and start transcription. So acetyl groups, acetylation, will increase transcription because they open up the promoters a little bit more, and then methyl groups will make things harder to get to. How I remember this: M for methyl, M for mute. So if I add a methyl group, I have muted that gene. We've kind of turned it off, or we've made it quite quiet. As opposed to acetylation, A for access. So if I have an acetyl group, I have increased the accessibility of that promoter site. We have more access, and so more transcription will happen. So let's say that we've got our acetyl groups attached to our promoter site, and that allows polymerase to jump in. So polymerase RNA, we had DNA polymerase in replication. Now we have RNA polymerase here in transcription. So RNA polymerase is going to bind to that promoter site, and RNA polymerase all by itself is going to separate the DNA double helix. No helicase is needed here. Polymerase can do it all by itself. So we're going to open up that DNA double helix, and then our RNA nucleotides, the A's use C's and G's. We don't have thymine in RNA, remember. We're going to add these A's, U's, C's, and G's in complementary base pairing. We have complementary base pairing, just like we do in replication. Oh my goodness, complementary base pairing. Just like in replication, we're going to have these complementary base pairs line up. We've got some weird vocabulary here. This piece of DNA is called the template strand, and we're actually going to add our new RNA nucleotides by following the code on the template strand, but the other guy is called the coding strand, which is opposite of the template strand, which is kind of weird, but here's the reason. So if I look at my template strand, T-A-C, T-A-G, when I code my RNA, we're going to get the complementary base pairs. So T to A, A to U, because it's RNA instead of DNA, C to G, T to A, I'm going to skip over here, G to C. They are complementary to each other. They don't match, they're just complementary. That's different from the complementary DNA strand. If I look at these guys, my coding strand and the RNA strand, they match with the exceptions of the A's and the T's and the U's. So otherwise they're a match. So because they match each other, because they're both complementary to the template strand, we call this the coding strand, because its code matches, even though I didn't use it to make the code. I'm also going to call that coding strand the sense strand, and then my template strand is the antisense strand, which doesn't make a whole lot of sense. See what I did there? But if you think about how this antisense strand is complementary to, not a match for, but complementary to the RNA, then it's a little bit more logical. All right, so here we have our template strand. Here we have our non-template strand, which is also called the coding strand, which is also called the sense strand. This is our antisense strand. So polymerase is going to start adding RNA nucleotides, the ones that are complementary to our DNA code. We're going to add these complementary RNA nucleotides, just like in replication, in a 5' to 3' direction. We're going to make some covalent bonds between those nucleotides. Those covalent bonds are a special kind called phosphodiester. You just need to know that they're covalent. So we're going to go in a 5' to 3' direction, just like in replication, for the same reason. And that reason is this. So my next DNA nucleotide is an A, which means that my complementary RNA nucleotide is going to be a uracil. So I'm going to have that nitrogenous base uracil bound to my 5-carbon sugar, bound to my phosphate group. But I'm going to start with those 3 phosphate groups that are bound to carbon number 5. Polymerase is going to use the energy bound up in those extra phosphate groups to make the new covalent bond between this guy and the next guy. So that bond is made using the energy from those extra phosphate groups. Those phosphate groups are on carbon number 5. So we have to add the 5 to the 3. We have to go in that 5' to 3' direction in order to make that new covalent bond, because the 5' end of the nucleotide is where we've got all that energy stored. Once polymerase has made its transcript, its messenger RNA from that DNA code, that RNA is going to start to move away from the DNA. It'll separate because it's a single-stranded guy. And once that RNA moves away, the DNA double helix will reform. And that's what's happening in this amazing electron micrograph. So we opened up the DNA so that we could make some RNA. Here, that DNA has closed back up. And so that double helix reforms after polymerase has done its stuff, and that RNA strand has released from the DNA strand. And you can see that that's actually going on at a couple different places in the picture. So either these are different genes that are being coded, or we started transcribing that gene here, and this is another guy, and this is another guy that are all working along the same chunk of. Transcription is going to stop when RNA polymerase gets to a terminator sequence. So we started at that promoter sequence. This is what attracted that polymerase to come and start doing transcription. And then we're going to end transcription at the terminator sequence. When we get to that terminator sequence, this is the stop signal for polymerase. Polymerase knows, well, we're done. Stop making the RNA. The polymerase will release from the DNA and go code for another gene someplace else. And then we've got our piece of messenger RNA here, ready to go make translation if you are a prokaryote. Again, if you're a prokaryote, that messenger RNA that you just made is ready for translation. We will go ahead and take that messenger RNA and use it to make the polypeptide that it codes for. Eukaryotes are a little different. What's going to happen is we're actually going to process that piece of messenger RNA first. So that messenger RNA that we just made using RNA polymerase in the DNA code, before we can send it out to a ribosome, we're going to process it. And that processing is called splicing. This is going to sound crazy weird, but we're going to take that messenger RNA that we just made and we're going to chop it up into a bunch of pieces. We're going to chop up that messenger RNA that we just made. We're going to take some of those pieces, we call those pieces the exons. We're going to take some of those pieces and we're going to glue them together. And then the other pieces we're not going to get glued in, and instead these guys are going to be recycled. The exons are the pieces that get glued together to make my mature messenger RNA that'll go to the ribosome and make a protein. The introns are the pieces that stay in the nucleus, and they're going to get recycled and turned into another piece of messenger RNA. How to remember this? Exons exit the nucleus, introns stay in the nucleus, and they get recycled. So that's splicing. The benefit of splicing, because it seems kind of counterintuitive to make all that messenger RNA and then just chop it up right away. The benefit of this splicing is that we can make more kinds of proteins. We have a lot of variability that we can take advantage of in terms of protein synthesis, because I can splice these guys up in different ways, glue those exons together in different patterns, and so I can take the same gene, transcribe it into the same messenger RNA, but then process the messenger RNA in different ways, and that will turn into different proteins. We actually make a whole lot more proteins than we have genes, which is pretty cool, and it's all because of this splicing. So we can splice the same gene in different ways to make different proteins. We're also going to add a poly-A tail. Literally, we just throw a bunch of adenines, poly-A, many adenines, poly, many, adenine A. We're going to add a bunch of adenines to the end of the 3' end of the messenger RNA. And the 5' end is going to get this 5' cap of a special kind of guanine. And that's going to allow the ribosome to bind to this piece of messenger RNA, which is what happens in translation. So translation is the synthesis of a polypeptide on ribosomes. Polypeptides are our chains of amino acids, and then we fold those polypeptides up into their secondary, tertiary, quaternary structures, and that's a protein. We can make a lot of the same polypeptide, a lot of the same proteins by taking advantage of this structure called a polysome. So what's happening in this picture? So this line here, this is our DNA. And then along that DNA, we've been making some strands of messenger RNA. So buried under all those blobs, that's messenger RNA. And on those pieces of messenger RNA are many ribosomes. Many, oh my goodness, many ribosomes. So many ribosomes make a polysome. This is a polysome. This is a polysome, this is a polysome, and again, and again. These guys are all using the same piece of messenger RNA to make the same protein over and over and over and over. So if there's a protein that we need a lot of copies of, we need a lot of one kind of protein, then these polysomes, many ribosomes on the same piece of messenger RNA can make that happen. I know that this is a picture of a bacteria because the DNA and the messenger RNA are together with the ribosomes. For us eukaryotes, the messenger RNA has to leave the DNA and go out of the nucleus to go find the ribosomes. We could still have polysomes, it's just not as cool because they're not also stuck to the DNA. All right, so let's talk about these amazing ribosomes that help us carry out translation. So ribosomes are composed of rRNA, that R stands for ribosomal. So we've got some ribosomal RNA and some protein together make up ribosomes. Ribosomes have two subunits, one large, here's a large, one small, there's the small, and together they make up a single ribosome. Ribosomes have binding sites for both messenger RNA and transfer RNA. We'll talk a lot about transfer RNA in the next couple slides. Ribosomes can be bound or free. Bound ribosomes are bound to the rough ER, and these are the guys that make the proteins that are excreted out of the cell. Our free ribosomes make proteins that stay in the cell. And then these ribosomes can be 70S or 80S. That S stands for Svedberg units, which is just like how far they sink in a test tube, which was invented by this guy Svedberg. 70S ribosomes are found in prokaryotes, those bacteria. They're also found in mitochondria and chloroplasts, and that's why we talk about endosymbiosis. Our 80S ribosomes, these are the guys in the cytoplasm of eukaryotes, so outside of the mitochondria and the chloroplasts. The amino acid sequence of a polypeptide, which can become a protein, is determined by the messenger RNA. And of course, the sequence of that messenger RNA is determined by the genetic code that is saved in the DNA. Messenger RNA strands that get translated are on average about 2,000 nucleotides long. But of course, we have some that are much longer, some that are much shorter. It just depends on the size of the protein that it's making. Here we have our messenger RNA. It is bound to the small subunit of the ribosome. Here's the large subunit. And then here are our transfer RNA molecules that are sliding in to that large subunit of the ribosome. We know what amino acids are coded for by these sequences of messenger RNA because we have figured out the genetic code, and then we organize that genetic code into a codon chart. A codon is a triplet of messenger RNA nucleotides. So if I'm looking at maybe CCA, this triplet or codon of messenger RNA nucleotides, I can look up in my fancy schmancy codon chart to figure out what amino acid is coded for by that. So I find the C and I find the next C, and then I'm like CCA. CCA is right here. That amino acid is proline. So I know that proline is coded for by this codon, this triplet of messenger RNA. Cool thing about our codon chart, this genetic code, it is mostly universal. Almost every living thing uses almost exactly the same codon chart, which is pretty cool because we can take advantage of that. And we'll talk about that later, the very end of this lecture, and then a lot in genetics. So let's talk some numbers really quickly. How many different codons can we put together? So we're going back to our fundamental counting principle. Codons are triplets. So there's 1, 2, 3 possible letters that I can put into my codon. There are 4 different letters that I can put in each one of those places. And so we get about 64, this doesn't account for repeats and whatnot, but 64-ish different codons. But there's only about 20 amino acids, which means that we have some codons that code for we have multiple codons that will code for the same amino acid. Multiple codons, same amino acid, like leucine. Oh my gosh, look at all these different codons. All these different codons code for the same amino acid. We call that degenerate or repetitive. We have these repeating codons that code for the same amino acid. That's known as degenerate. And it's kind of nice because if I have a tiny little mutation in my DNA, which means a little mutation in my messenger RNA, I can still get the same amino acid, and so my protein is not affected by that mutation. Let's practice using our codon chart. So if my messenger RNA strand is this, let's figure out what our polypeptide chain will be. So AUG, how we read our codon chart. We're going to go A first, U next, and then I find the square where those two intersect, and then I'm going to find G last. AUG is right here, and my amino acid is MET, which is short for methionine. This MET is almost always our start codon. So sometimes we refer to this as the start codon, methionine or MET. Next up, CCC. CCC is pro, that's proline. UGU, UGU is cysteine, that's the special amino acid that has sulfur in it. GGA, GGA, that looks like glycine. GCC, GCC is alanine. And then we have UAA, UAA. Ooh, that's our stop codon. So the ribosome would know to stop making a polypeptide sequence at that stop codon. Let's do another one. This time, I'm giving you the DNA instead of the RNA. So we're going to have to do some transcription first to make a messenger RNA, and then we will translate into our polypeptide. This is the antisense DNA, which means that it's the complementary piece of DNA, not the matching. So we do need to find our complementary base pairs. If I have a T in the DNA, we're going to have an A in the messenger RNA. An A in the DNA, it's going to code for, remember, not a T, but a U in RNA, and then a G. And then ACA, GGA, it's going to turn into CC. Remember that A goes to a U instead of a T. ACC will be UGG, TTT, AAA. And last but not least, we have ACT is going to turn into UGA. So that's our messenger RNA that we made in transcription. Now let's go ahead and do some translation to turn that messenger RNA into a polypeptide. AUG. We're going to go AUG. Once again, we have that methionine, our start codon. Next up, we've got ACA. ACA, threonine. CCU. CCU looks like a proline. UGG. UGG, tryptophan. AAA, lysine. And then we've got UGA. Hey, it's another stop codon. We have our stop codon, and so our polypeptide chain would stop being made. So you probably figured out already that we have a whole lot of complementary base pairing going on here. So our messenger RNA and our tRNA are going to be complementary to each other, just like the messenger RNA was complementary to our DNA. So the triplets that we were just talking about in our messenger RNA are called codons. So there's a codon, there's a codon, there's a codon. On the transfer RNA molecules, we have also triplets, and these are going to be called the anticodons because they're complementary. A to U, C to G, G to C, in all of those pairings. So complementary base pairing between the codons and the anticodons of the messenger RNA and the transfer RNA. So how do we even get those amino acids onto our tRNA molecules? With an enzyme, of course. We have an enzyme whose name is tRNA activating enzyme, and its job is to activate transfer RNA by adding an amino acid molecule to it. So how this happens, we start with some ATP. It's triphosphate because it's got 1, 2, 3 phosphate groups on it, which means lots of energy is bound up here. That's going to bind to one part of the enzyme and give it some energy. We have an amino acid that's going to move on to one piece of the active site of the enzyme. Transfer RNA is going to move to another part of the active site of the enzyme. And we're going to use that energy in ATP. It actually is going to lose not one but two phosphate molecules. Phosphate groups. So now we're down to just one. This is a MP, adenine monophosphate. Only one phosphate group is left. And the energy from that molecule of ATP that we turned into AMP is the energy needed to glue together the transfer RNA and the amino acid. So now we have our charged tRNA. This enzyme added the amino acid to that molecule of transfer RNA. So now that we've got our activated or charged molecules of tRNA, we are going to start some translation. So step one, our messenger RNA is going to bind to the ribosome's small subunit. So messenger RNA is going to bind to the small subunit. Whoa, that's too many L's, small. And then the large subunit will come and sit on top of it. We're going to start at that 5' cap. We've got our start codon that's going to code for methionine. It's going to be right here. The next thing that's going to happen is some transfer RNA is going to line up with the codons. And we're going to do that on the large subunit of the ribosome. We have large subunit is here. Small subunit is here. There are some special binding sites on the large subunit called E, P, and A. We have E, P, and A. The first amino acid is actually going to start in the middle in the P site. So the first guy is going to go to the P site. Amino acids look like this. They're going to do that complementary base pairing, codon to anticodon, there on the ribosome. First guy in the P site, now a second transfer RNA, complementary to the second messenger RNA codon, is going to bind to the A site. So we have a P site and an A site, transfer RNAs in each of those places. And then the ribosome is going to use energy to move all of the amino acids that are on the first tRNA onto the amino acid of the second tRNA. So we're going to take this guy and chunk it onto this guy. This guy gets chunked onto that guy. And then it looks like this. This guy that used to be here is now attached to all the guys that used to be there. They've been moved over. So the amino acids that were on the P site tRNA are now attached to the amino acids on the tRNA of the A site. And then, because this tRNA no longer has an amino acid, it's going to slide over to the E site. And then from the E site, it will exit. E for exit. That's going to allow this guy to move from the A to the P. When P moves to E and exits, A can move over to P, which means that we can then add another transfer RNA to the A site. The ribosome will take all of those amino acids and chunk them onto that guy. And then this guy will move over and exit. This guy will move over, and then we do it all over again. And we do it again until we get to that stop codon. And here's the map of the genetic code. So this process of translation, the codon, anticodon, amino acid groupings is pretty much the same for all living things, which means that we can do this. I can take a human cell and I can take its DNA and I can chop out a gene, perhaps the insulin gene. I can also take a plasmid, which is a small circular piece of DNA, in bacteria, and I can cut it with the same enzyme. And so this sequence that got cut is a match for these cuts. And then the insulin gene will stick to and mash itself into the middle of that plasmid, that little circle of DNA. I can then blast that circle of DNA back into a bacteria. Bacteria can make babies just about every 20 minutes. And so after a couple days, I can have this huge petri dish or perhaps even a flask or a vat of bacteria that all have the gene to make human insulin. And because the code is the same for bacteria as it is for humans, pretty much, the bacteria are going to use this gene to make insulin. The human insulin gene in bacteria will form insulin protein molecules in the bacteria. And then I can extract that insulin from the bacteria and sell it to people at grossly high prices, and people with diabetes can be treated, which is pretty darn amazing. So we achieved all of our objectives and we almost did it in under 30 minutes. I'm so proud of us. We defined transcription. We described the process of transcription, that polymerase that is going to use complementary base pairing to add RNAs with phosphodiester covalent bonds in a 5' to 3' direction. We outlined some of those processes involved in gene expression. We talked about methylation and we talked about acetylation, which can either mute or give access to those genes. We also talked about introns and exons, gene splicing that allow us to rearrange some of the pieces of messenger RNA. We then defined translation using our messenger RNA code to make a protein. We talked about the structure of the ribosome, large subunit, small subunit, ribosomal RNA and protein. We used our codon chart. We described how translation happened, those EPA sites, and then super shortly talked about genetic code is universal. Well done, my friends.